Welcome to everyone to today's MPG Primer. We are very excited to have Dr. Mehrtash Babadi here today. He is the Director of Computational Methods in the Data Sciences Platform at the Broad Institute of MIT and Harvard. He leads the Cellarium Lab, a research group dedicated to developing computational tools and services with strong emphasis on innovation, mathematical rigor, usability, transparency, and reproducibility. Cellarium Lab's recent activities include developing probabilistic machine learning models for enhancing and interpreting single-cell droplet-based and spatial omics data, cellular morphology data, electrophysiology data, and voltage imaging data with broad applications to psychiatry, cardiovascular, and infectious disease research. They also do precision quantification of raw measurements enabled by modeling epiphenomena and experimental artifacts on par with biological phenomena. And finally, they also have a focus on learning the geometry of massive scale multimodal single cell measurements and building a large foundational model of cells using self-supervised contrastive and generative AI techniques. There is a lot in there that your group does.
There's a lot in there. Yeah.
And we are so excited
Who wrote that?
It just came from the sky. Right. We are very excited to hear all about your group today. And would you like questions during your talk or at the
yes,
end?
please feel free to ask questions. Yeah.
Perfect. And so the mic is already on, so feel free to go up and just speak. It'll turn on automatically as you talk. Or if anyone online has questions, just type in the Q&A. Thank you so much.
All right, pleasure. Okay, it's a great opportunity and wonderful to present to this community. I'm Mehrtash, and thanks so much for the generous introduction. All right, so let me start by just showing you the faces of the wonderful people that I have the opportunity to work with every day. We are the Cellarium Lab in the Data Sciences Platform. And very generally, what we do is that we're developing machine learning models and tools and services that are generally inspired by analysis challenges and unrealized potentials of all the cell biology data that we're collecting. And that includes different types of cell biology data modalities, including single-cell omics, voltage imaging, morphology profiling, and spatial molecular profiling. Just a quick background about the revolution of single-cell biology. So as you know, before 2015, the only tool that we had for looking at the transcriptome was doing basically analysis at the level of bulk. So we would basically just take a tissue, put it in a blender, and make a smoothie out of it, and then we would just get an idea of the average expression from all the cells in the tissue. And the advent of the single-cell technology is the thing that allowed us to basically look at the heterogeneity of gene expression across tissue. So basically, instead of looking at the tissue like a smoothie, we're looking at it as a fruit salad. Now, I don't know where I stole this analogy from, but I've certainly heard it from Aviv. Yeah, so and ever since the advent of the technology, it has led to a rapid rise of single-cell atlases, and just a number of the names of the major atlases. And across all these atlases, we have generated hundreds of millions of cells, and this is an effort that is not stopping anytime soon. If anything, it's accelerating. So what we've been doing for a good while was, and still is, in part, to develop computational tools and methods for doing different things with single-cell data. That includes enhancing the data, that includes inferring something from the data, dissecting the data, decomposing it into something that it is more understandable. And here I'm just showing a couple of highlights of a couple of tools that we've developed over the years. And I'm going to just very briefly overview a couple of them. But that's not really the focus of my talk. So I will just keep it really high level. And I encourage you to either visit our website or look at the papers if you're interested in more details. OK, so today I'm just going to talk about 3 of these tools very, very quickly, starting with the latest model that we've developed, which is called CellcCAP. So this is developed in collaboration with Stephen McCarroll's lab. And the work is spearheaded by Yang Xu and Stephen Fleming. Okay, so what problem does CellcCAP try to solve? So CellcCAP is a model that helps us make sense of single-cell perturbation data set. So what is a perturbation experiment? The perturbation experiment is that you basically just take a bunch of cells, and then you treat them with in different conditions or with different perturbagens. And so let's say that this is the distribution of the state of your cells before any perturbation in some representation. This dimension 1, dimension 2 could be gene 1, gene 2, or it could be like PC1, PC2, or whatever representation that you like to visualize your diversity of your cellular states in. Okay, so by imparting some perturbation, let's say perturbation A and perturbation B, these cell distributions evolve, right? So there are a number of questions that we would like to be able to answer. One of them is that, how do individual cells respond to perturbation? Because that's the thing that we never observe. We only observe distributions of cells. So sort of trying to infer how things would have looked like at the level of single cells, such that if you applied that perturbation to individually to all of these control cells, you would get basically that distribution of the perturbed cells. That is like one thing that we would like to be able to do. And the reason that this is an interesting question to answer is because then you would know which cell states respond to your perturbations, which cell states do not respond to perturbation. What are the markers of strong response? What are the markers of weak response? The other interesting questions that you would like to answer is that, okay, so fine, we know that how each of these cells respond to perturbation, but what is the nature of this response? Can we sort of decompose it into sort of more elementary, more understandable response programs, and then sort of be able to then ultimately recompose these perturbational responses in terms of like, some of these elementary responses. And then ultimately, we would also like to know that, okay, so here I'm just showing only 2 perturbation conditions, but imagine I have like thousands of perturbation conditions, right? We would like to know how these perturbation conditions are generally related to one another. Is it that, for example, knocking out KLF1 is very similar to like, knocking out some other gene? And sort of being able to have an embedding of your perturbations in a way that you can say that, oh, these 2 perturbations are similar and those 2 perturbations are dissimilar is another thing that we want to do. And these are some of the things that CellcCAP enables us to do. Please.
Quick question, 2 questions. What's the minimum number of cells would this tool, what's the resolution, minimum resolution? Because there's always an issue.
Yeah.
Especially, let's say, when we are looking at eye diseases, we don't have enough sometimes, there's not enough for us to work on. So what's the resolution there? And then looking at individual cell level, which have genes, what's the resolution there? Like how many genes should respond in a particular cell for you to know it's being perturbed enough?
Well, these are hard questions. And I don't have answers to that, but I can give you like rough answers. So if you have hundreds of cells, that should probably be good enough for this to work. At a level of hundreds of cells, we probably don't even have a good idea about what's the cell state heterogeneity. So being able to break down responses to individual cell states, I think it's a little bit big ask. But I think you would start to get like reasonable results starting at like hundreds of cells. But I think once you're at the level of like thousands of cells per condition, I think that's where you really start to be able to resolve the cell state heterogeneity and sort of also being able to have an understanding of the cell state specific responses. In terms of number of genes, that also completely depends on the nature of those genes. If these are like very highly informative genes, then I think you can get away with as little as, let's say, 50 genes. But if these are like just random bag of genes, then probably they're not very informative, and then you need more of those. Yeah, but ultimately you need just to try the tool to see if it works. Yeah. Okay, so how does CellcCAP work? And these are the wonderful people who developed it. The idea is this. So for those of you who are familiar with variational autoencoders, it's effectively a variational autoencoder with a bunch of additional structure. So what it does is that it takes a single cell's observation together with its perturbed conditions. Like basically, the tool takes as input during its training that here's a cell, here's the state of the cell, and here's how the cell was perturbed, what perturbation it received. Then there's this encoder neural network that the job of this neural network is to be able to predict what the state of the cell could have been before perturbation. So it sort of tries to guess, where the cell could have come from. And then there's this other piece of this whole plan, which is something which we call the attention mechanism, which is a building block of a transformer architecture. But this is basically a mechanism that allows the perturbation that the cell has received to attend to the state of the cell, and based on this sort of the level of attention, sort of propose that how the cell would then respond to the perturbation. And then the two effects will be summed, the pre-perturbed representation of the cell together with the effect of the perturbation, and then ultimately this is recomposed into what we believe is the state of the cell, and this is ultimately decoded back to the gene expression state, the gene expression space, and sort of ultimately connected back to the original state. So basically, in a shorter version of this is that you take basically an observation, which is the perturbed observation. The model tries to break it apart into the initial state of the cell and the effect of the perturbation, adds them together, and then decodes it back and tries to match with the original observation.
I'm just curious for when you said that it's tried to learn pre-perturb. Usually we do have wild type. We always have wild types. So why can't we start with known rather than do a prior set? Here it is, what's the wild type, learn from it, and here is the perturb.
Yeah, we know the wild type in distribution, right? So you don't know exactly. So you have like a cell culture in without any perturbation, and you have like another culture that you have done a perturbation. These are different cells. These are not the same cells, right? So this is a counterfactual. Like if this is like a perturbed cell, which unperturbed cell it could have looked like before perturbation, such that after perturbation, then it would look like this, right? So because these are not the same set of cells, so you need to connect cells across different data sets.
So did you ever tested this like in lab to actually confirm how further apart that delta is from? Because you're saying, I understand they are not the same cell per se, because there's one media wild type and there's a different media, even different cells. But did you look into that? Because right now, for example, base Prism and all, they're using bulk RNA-seq and single RNA-seq to do study together. So what is already there making use of that? So that's what this question was coming from.
Yeah, so we have, yeah, I would definitely, would encourage you to look at a preprint. We have done a lot of testing using simulated data, like lots of different simulation environments. And we have also tested it in 2 real data sets. One of them is like monocytes that are exposed to different pathogens, and the other one is combinatorial perturbations on CRISPR-A perturbations on cell lines. And so we have like we haven't gone back to the lab to verify every one of our findings, but it makes a lot of sense, the results that we see when we apply to like real data. Yeah, but that's actually a great thing. So if you to just be able to go back to the lab and maybe purify your cell state a little bit and then just apply the perturbation on that and see whether it's really compatible with the thing that the model is telling you. Well, you're asking lots of wonderful questions, which means that I'm going to skip a lot of my slides. So the most important thing about CellcCAP is that it actually ultimately has a transparent model. Like all the things that it learns and all this variables that it sort of learns, these are like very easy to interpret. Like these are like transcriptional programs. Like these are like the relevance of each transcriptional program to each perturbation condition. These are the attention rates, like how much do different cells, use different transcriptional programs in building up their response and so on and so forth. So this allows, basically the model to be like less like a black box model, but something that more like an open box model that you can just take a look at the results and learn something from of biology. Just show you just 2 quick results. Okay, on the top, these are like the monocytes that were exposed to pathogens that I mentioned. I think these are like 1, 2, 3, 4, 5, 6, 7 pathogens, and then you can, based on the results from CellcCAP, you can group different pathogens based on how they impact your monocytes. This is like a more this is another experimental setup that is CRISPR-A perturbations, like K562 cells, that we have a lot more perturbation conditions. But again, you can see that based on the results from CellcCAP, you can group different types of like CRISPR-A perturbations to clusters that result in the similar transcriptional responses in cells. But the most important thing is what I mentioned about the interpretability of the result from CellcCAP. So this is like a typical thing that you see from CellcCAP. So you have here on the conditions on the y-axis and transcriptional programs that CellcCAP learns as building blocks of the response on the y-axis. And for example, this is telling you that this perturbation condition, let's say 24 hours after like infection by this pathogen, your cells generally responds in terms of like this program, and these three programs, and these three programs. But these other three programs are not used in the response. And then you can go back and ask that, okay, so which cell states actually specifically responds strongly to this program, to any one of your programs, right? So not only you can dissect perturbational responses in terms of like building blocks, but you can then go back and ask which cell states is more likely to respond to these perturbations in that specific direction. And then you can then ask what are the markers of those cell states, and then you can ask, okay, so what is actually the nature of that response, and so on and so forth. So it's actually pretty easy to interpret the results. Okay, so I like this one. This is cool. So I'm gonna go through this one too, but then I'm gonna actually jump to my main topic. So this is another tool that we developed. It's called CellMiner. So it solves a problem in a totally different domain. In the domain of optical electrophysiology. So the work is spearheaded by Bryce Wang in my lab, and it's a work we did together with SAMI, optical profiling platform, spatial profiling platform now. Okay, so what problem are we trying to solve? So patch clamp electrophysiology of neurons is the gold standard way of looking at the electrophysiology of your neurons. The problem is that it's a very laborious technique. You need to go to the lab under a microscope, patch your individual neurons. Your patch might fail, but ultimately, if everything works, then you can get really good data. Then, on the other hand, we have these optogenetics-based techniques. One of them is called OptoPatch, and there are like any number of them. That basically, the idea is that you use fluorescent reporters to optically measure the electrical activity of your cells. And also you have like these ion channels that you basically transfect your cells with so that they express those ion channels, but those ion channels are light gated. So you can then stimulate, you can basically shine a specific laser on your cells, and it basically opens the ion channel. So that way you can sort of inject current inside your cells. So you can do the good old electrophysiology experiments completely in an optical way. The problem is that it's a very noisy data generation platform. It's very high throughput. You can look at hundreds of cells at a time, but the data looks like that, super noisy. So we ask ourselves, can we kind of marry the two, sort of like have the high throughput of optical electrophysiology, but have sort of data quality that resembles the gold standard data quality that we get from patch clamp electrophysiology. So we used a cute technique to solve this problem. So refresher. I guess most of you at this point have heard about large language models and how they model language. It's done by playing a game. You basically take a piece of text, you mask some entries, some words in that piece of text, and then tell the model what are the surrounding words, and then the model has to guess what is the masked word from the surrounding word. So let's play this game on a movie. So let's just take your optical electrophysiology recording, which is a width by height by time. It's a three-dimensional cube of data, right? And imagine this is a sentence, and mask a couple of pixels here and there in this spatial temporal sentence, and then train a massive neural network to predict the intensity of those masked voxels from the rest of the voxels. Okay? Now something very cool happens. Well, noise by definition is not predictable. So you can never predict noise, but signal is predictable. So which means that the best that you can ever do is to predict a signal, and you can never predict the noise. So as simple as that, once you train this predictive model and then just run this predictive model on your data, it separates your data into noise and signal. So if you don't believe me, let me show you some results. So this is like a raw movie frame. You can barely even see a neuron here. And here's what happens to it after playing that game on it. Now you can actually see the neurons. Here is like the sort of the raw fluorescence trace of 1 pixel, in time, super noisy. But now here's like what you see after denoising. And here's even better. So on the top, you see the raw data. On the bottom, you see after denoising. So you can clearly see the difference. So it's a cute trick, right? Simple, cheap way to enhance your data. I'm going to skip this one, unfortunately, even though I love this project, because we don't have time. You know, actually, let me explain it in 10 seconds. What about that? OK, this is a method still under development. Oh, finished development. We're writing a paper. Work is done by Sandeep, who's a PhD candidate in Fei Chen's lab. But here's the problem we're trying to solve. So when you have like bulk RNA-seq data, it's very easy to do differential analysis. Bunch of genes here, their expressions, bunch of genes, their expressions, you're good, right? When you have single-cell RNA-seq data, it is also easy to do differential analysis. First, you cluster, slap a label on your cells, and then maybe compare different cell clusters in terms of their gene expression with each other. At least we know some ways of doing differential analysis. But when you have spatial transcriptomics data, like a bunch of images, condition A, a bunch of images in condition B, what does it even mean to compare, to see like what is different between these two images? There's so many things that are different between these two images, right? I mean, even if you just take the same, just take an adjacent slide from your healthy sample, the same specimen, it's gonna look different. So even like making this a well-defined question, like what does it mean to do differential comparison between tissues is a philosophically difficult question to grasp. And sort of we try to take like the first step in that direction. But the short version is that imagine, so the reason that it's a difficult problem is that we don't have a basis. Like when we're dealing with gene expression data, we have a basis, genes. When we're dealing with single cell data, we have also a basis, cell type and genes, right? So we have a way of like canonically building groups for doing comparison. So we also need to do that for the spatial data. So the idea here, and this is by no means the best solution to the problem, but a solution to the problem, is basically to take your individual tissues, break them apart into thousands of small patches, okay, and learn a common space onto which you can embed these patches on. So that you could say that, oh, these patches are coming from this image, these patches are coming from that image, and so on and so forth. Which patches are used more in one condition, which patches are used in less in condition? What is a prototype? What is the hallmark of a disease? What patches are overrepresented in a mouse lung that is injured or something, And then once you identify like these sort of motifs, then you can go and do some enrichment. You can say, okay, I only want to look at these specific regions and these specific regions that have enrichment of a certain motif, and then I want to do my differential gene expression analysis specific to those motifs. That's kind of like the way it works. Skipping the details of how we actually learn those representations of motifs, but let me just show you like one interesting result that we get from that. So one of the cool things about learning an embedding or a representation of your tissue motifs is that it gives you a magic wand, which means that you can pick one region in the tissue and you can ask what other regions in the tissue looks like that. The way you would do it is that it would just take the patch of interest, embed it, put it in that space, and take every other patch, also embed it in the same space, and then see which patches are similar to it and which patches are dissimilar to it. So here's just a couple of examples. So if you choose just this patch, we can then highlight these patches. If you choose like this, this blue patch, we just get these blue patches. If you pick this sort of disordered red-blue patch, we can highlight these red-blue patches. So this already shows that the representations that we learn have an understanding of histology. It knows that, oh, these are like tubules. These are the different types of tubules. These are like disordered tubules. And then we can then take the next step and then try to establish a correspondence between the representations of these patches and the gene expressions of cells that make these patches, and then identify which cell states might be tuned to specific tissue microenvironment, and so on and so forth. Okay, main topic. So this is the thing that we've been working on. We call it Salarium AI platform. So what is it? What is it? It's something that we're building sort of really out of necessity because we started a couple of collaborations. We want to do some analysis on 10 billion cells. We don't know even how to store 10 billion cells. We don't know how to even query 10. We don't even know how to do that for 100 million cells. So we really needed to build a platform so that we can do massive scale single cell analysis so that we wouldn't need to worry about scale, right? So we could basically just take the same analyses that we've been doing before and just run it at whatever scale. So just by way of example, so these are some of those collaborations I mentioned. This is sort of like an overview of the human brain variation project, co-led by Steve McCarroll and Evan Costco. The idea is to take 200 human brains-ish, and then take like dozens of brain regions from each donor, and then do spatial transcriptomics and multi-omics and everything. So it's going to produce something of the order of like 100-ish, 200 million observations. And then once we have all of this data, we probably want to do some type of a covariation analysis, identify cell populations that covary with age, with sex, with disease, condition, or whatever the label that we have over these donors, so that we can identify some interesting new biology. Or maybe the other thing that we want to do is that we want to, since this is like spatial transcriptomics data, we want to take tissue purifier, the algorithm that I described earlier, and run it at the scale of this data set, sort of to learn like global embedding of brain tissue motifs. Okay. But to be able to do these things, we really need to have a software infrastructure that allows us to store all of this data, query this data, train models on this data, and so on and so forth. So this is another project still in the making. This is led by Anna Greca, but this is a project aimed at the space of rare diseases. And this is going to produce about 10 billion cells. So even like 2 orders of magnitude more data. Okay, so what is Salarium AI platform? So this is sort of the cloud native software that you're developing that allows us to sort of scale up single cell analysis. And by scaling up single cell analysis, I don't mean to run a pipeline on the same small single cell data set again and again and again. It basically means that if you want to embed like 2,000 cells, and you can do it on a laptop, and now if you want to embed 200 million cells, you can do basically just that. Like instead of like chunking the data into pieces and just looking at each data piece separately, integrate all of that and do like this, the same analysis just at much larger scale. So the components of the AI platform is that there's a data store, and this is sort of a cloud-based database that you can just put your data in and get your data out of it. And then there's a machine learning library, and these are like implementations of machine learning algorithms that are scalable and distributed, so they can basically just hook it up with this data store and then train models directly. They're microservices, and these are basically components that we use very often, like building nearest neighbor graphs. We always want to build nearest neighbor graphs, so it's good to sort of refactor it so that you can then give it like 100 million cells and say, like, build a nearest neighbor graph from these 100 million cells for me. And then ultimately, we can combine these things to build applications. Interesting applications. So I'm going to skip some of these details and just directly go to applications. So one application, the first application that we're building is called CAS, or Cell Annotation Service. So what problem does it solve? It solves the problem of being able to search. So, and here's what I mean by that. So one of the very first things that you want to do in biology is you want to discover a mechanism. You want to see like what is different between this condition and that condition. So to be able to do that, you need to identify which studies are similar to your study, if you don't have your own control, and which study is not similar to your study, so you can sort of build cohorts. So that is that requires you to search, like which cells are similar to my cells, which cells are not similar to my cells, and then slice and dice it sort of to build your cohort. This is basically like a search engine in the way that we search internet texts and stuff by building indices and then doing the search over these indices. And here's exactly what we're doing. So we take lots of data, lots of reference single cell data from atlases, and using our like super scalable machine learning library, we can embed all of them into compact vector representations. And you can think about these as sort of like signatures of each of these cells. And now if you have like a new cell, you can match your new cell with all these reference cells based on their signature and sort of nominate who are the similarity hits.
How do you generate these low-dimensional representations at such a large scale?
Yeah, great question. So the question is that how do we generate these low-dimensional representations at such a large scale? Well, we have scalable algorithms. So that's actually what but so basically we're using stochastic algorithms like stochastic gradient descent or like specific implementations of the good old like embedding algorithm like PCA. Like PCA, we've implemented a version of that basically scales to all data sizes.
But as you add more cells, do you not have to then like recompute, or can you plot them down to the same space? Even though
you can plot them at some point, it stabilizes, like
Right.
because once you sort of exhaust the diversity of all the cells, it sort of reaches a plateau. But you can also incrementally always retrain as you get more data. Yeah, sure. Okay, and so at the moment we have trained it on 86 million cells, all the cells that you have in the cell by gene repository, and so like I said, it's a search engine. You give it a cell, it tells you who are the similar cells. But one simple thing that you can do with it as a first application of the search engine is that you can just take the cell type annotations that authors have provided over those cells. Whatever people have provided. Most of them are probably wrong, but well, most of them can be wrong. Most of them are right, but some of them are wrong. So if you just look at the wisdom of the crowd, hopefully the crowd is not too wrong, which means that by just looking at the consensus cell type annotation that the community refers to cells that look like this, you can come up with a good cell type label. So that's one thing that I'm showing here is that you give it a single cell data set and it just generates labels for you by doing the search. We are releasing this publicly either by the end of this month or next month. So you should go to our website, which is not launched yet, but it will launch next week. It's called Salarium.ai. And then there's a sign-up form that you can register to get sort of early access to Cell Annotation service. And then we would love for you to use it and give us feedback. Okay, so search is an easy problem. The harder problem.
Sorry, like you just mentioned, the annotation may be wrong. So are you correcting them first and then putting it in there? Or are you assuming?
Not yet, but that's planned. Yeah.
And when you say similar, so there is similarity and there is identical, right? So similar is, I had perturbed my cell at 1.5 hour and it was 3 hours, they look similar. Is that the similarity? Is it 70% similar if I think a linear? What does that really mean?
Very specifically, similarity based on gene expression pattern. Like two cells are identical in our view, in this view, if they have the exact same gene expression. But the more their expression values across all genes vary, they're like less similar. But I guess to your point, you can emphasize on some genes more. How do you choose like how to scale different genes? Like, do you want to pay as much attention to mitochondrial genes as like transcription factors? Probably not. So that's why in Cell Annotation service we have different models that are like trained for different purposes. So we have like very generic models, we have like more fine-tuned models, and then we are hoping that over the years we just keep adding more and more models for like more specific tasks. I'm running out of time, but this is very cool. So let me just finish this part. But okay, so this is not the reason that we doing search was a side effect of building Salarium AI platform. That was not the goal. The goal was this. So we kind of had this vision that we're as a field generating so much data that give it 5 years, if not give it 10 years, if not give it 20 years. We have probably generated so much data across so many different conditions that even the most complex and contrived questions that you might ask, like I'm going to take this cell with this genomic variations, in this metabolomic context, in vicinity to those cells, and yada, yada, yada. And then I want to do this perturbation, I will wait this many hours, and I want to see how my cell responds to that. So even if you ask a very complicated questions like that, the answer to this question, different bits of pieces of answer to this question probably lies somewhere in all of the data that you've generated. You just need to integrate all of that into a whole. This is exactly like how ChatGPT works. It gives you answers to your questions, generic questions, but the specific question that you're asking, that question doesn't exist on the internet. Similar questions do exist. And the idea of integration of data using AI or ML is exactly that. That basically you will learn how to piece together pieces of answers, to just basically be able to answer like novel questions. Okay. So the roadmap to doing that is that we want to take all this data that we have, we want to train the so-called foundation model, a model that really understands the grammar of all the data we've generated, and then fine-tune these models for specific tasks. Like some of those tasks could be that I've measured this gene expression of this cell, and tell me what is the high-C contact map of this region of the genome to some other gene of the genome. I have a cell that it's initially in this state. I want to perturb it. How does it respond to perturbation? And to be able to build this specific technology, we're using transformers. And transformers are basically the same thing that we have used in natural language processing. I'll be done in 1 minute. Yes. Yeah, and I'm going to skip this slide, but basically, all I'm trying to say is that transformers have this internal structure called self-attention, and it's a way of different words to attend themselves. And that building block is also very relevant to cell biology because, it allows us, to just model interactions between all the biomolecules. Basically, it allows us to model biochemistry. So Salarium GPT is basically the name of this tool that we're working on, which is basically like an engine for answering arbitrary queries about your cells. So here's like one example. Like imagine like you have a bunch of T cells, and then you can ask that, I know that my you don't even know it's T cells, by the way. So you can say, I have a cell. That cell is IL-32 positive, CD3 positive, and IL-7R positive. What cell could it be? Or like, based on just this limited information that I'm giving you, and given that these are like your possible options, tell me like what this cell could be, and then it sort of can highlight it for you. And then you can give it more information. You can say, oh, by the way, this cell is also CD2 positive and CD4 positive. Now tell me like what it could be, and then it sort of narrows it down. And then you can say, oh, by the way, it's like LGA-LS3 positive, so it's an exhausted cell. Or like it's KLRB1 positive, so it's an effector cell. We know that, but the model also knows that. So that's sort of the premise of Cellarium GPT. You can just give it factoids, and the more factoids you give to it, the better of an understanding it will have of what the cell state is that you're looking after. Okay, so I think the longer-term vision of the stuff that I just said in the last slide is to extend it beyond cell phenotypes and sort of marry it with structural biology and ultimately take steps towards like building cell language models, like comprehensive models of cells that then you can interface with natural language models and then use it as a very generic purpose ChatGPT for biology. Check out our website, Cellarium.ai. Yeah, it looks kind of like a startup, but it really is not. It's just, yeah. Anything.ai is a startup these days. Thank you so much.